This is a lecture course on synchronization of oscillations, Part 2, Time Scales. All self-oscillations can be characterized by the following parameters: shape, amplitude, time scale. Of these three parameters, only the time scale is relevant to synchronization. In the figure, one can see oscillations of the blood pressure of a laboratory rat. When we analyze synchronization, we usually ignore the amplitude and the details of the shape of such oscillations and pay attention only to the time scale, which is a parameter that characterizes how fast the quantity being observed roughly repeats a certain pattern. The most general definition of synchronization is an adjustment of time scales of self-oscillations as a result of interaction between them. In particular, in most cases, before the systems start to interact, they oscillate at different time scales. With this, after they start interacting, as a result of some coupling between them, and after all transients die out, their individual time scales are changed anyway. However, we say that synchronization took place only if their time scales become either equal or rationally related. This means that the ratio of their time scales should be equal to the ratio of integer numbers. Let us illustrate synchronization in living cells. In this slide, the left-hand movie shows calcium oscillations in a single heart cell. A number of such cells were allowed to form a cluster, and they started to interact. Calcium oscillations in the whole cluster were recorded, as shown in the movie to the right. When many cells demonstrate a splash in the calcium concentration simultaneously, their total activity increases, and this is shown as a splash of warm color in the movie. So far, the concept of a time scale was introduced intuitively. This is something to do with a number of splashes of activity per time unit. However, to characterize synchronization quantitatively, we need to specify exactly what the time scale is. It is easier to do so for perfectly periodic oscillations, from which we start. The figure shows a recording of some quantity measured in an experiment as a function of time. A periodic function of time is defined by the equation in the box, which says that for any value of time t, the value of the function is equal to its value separated by time interval T. I emphasize that this condition must be satisfied not at some selected times, but for any time t. The time interval t is called the period of oscillations, and the inverse of the period is called the frequency of oscillations. If oscillations are not periodic, no single period can be introduced. The time intervals between two successive splashes of activity will vary from one splash to another. Here we can introduce the so-called instantaneous period Ti, which is the time interval between two successive maxima of our function of time. Instantaneous periods will vary from one splash to another, so we will have a sequence of values of Ti, where I is an index counting maxima. We can also introduce instantaneous frequencies, Fi, as inverses of instantaneous periods. However, it might not be convenient to operate with many periods. We would prefer to have just one number to characterize a time scale. We can obtain this number by averaging all instantaneous periods over time and ending up with an average period t and with its inverse average frequency. Alternatively, we can consider most typical period and the respective most typical frequency. To obtain most probable period, we need to estimate the probability density function from instantaneous periods. As an example, consider the beating of a human heart. This picture shows the so-called electrocardiogram, a signal that measures electrical activity of the human heart. We know that our hearts beat rhythmically, but it is also very well known that time intervals between successive heartbeats vary considerably. One can introduce instantaneous periods as the time intervals between the sharp high peaks in the ECG. Once these periods are recorded and stored on a computer, one can estimate their one-dimensional probability density function P1, also called a distribution for brevity. To obtain the distribution, we find the minimal and the maximal values of instantaneous periods and split the whole range of variation of periods into boxes of size delta t. We then count the total number of periods which fall into each of the boxes and divide this number by the total number of periods recorded. In the figure, two distributions are shown for the same human in different physiological states. Start from the shaded green curve, which illustrates one of the states. The location of the maximum of distribution points to the most probable period. In this case, the mean period coincides with the most probable one. In the second case, illustrated with a red curve, the most probable period corresponds to the highest of all peaks in the distribution and is shifted to the right as compared to the first state. With this, the mean period remains almost unchanged and does not coincide with the typical period. What happens when two systems are coupled and interact? Their individual timescales become affected by each other's presence. Moreover, instantaneous periods get adjusted during interaction. I would like to illustrate a special case of a unidirectional coupling in the human cardiovascular system. It is well known that breathing strongly affects the heartbeats in humans, but the influence of the heart on breathing is negligibly small. This video shows simultaneous recording of the human electrocardiogram and breathing. When the subject breathes in, the breathing signal goes up, and when he breathes out, the breathing signal goes down. One effect from breathing on the ECG becomes apparent instantly. The amplitude of ECG decreases when the subject breathes in and increases when the subject breathes out. But there is also another, more subtle influence. If you look closer, you will notice that when the subject breathes in, the heartbeat intervals become smaller, which means that the heart beats faster. And when he breathes out, the heart slows down. So the presence of rhythmic breathing modulates the heartbeats. Modulation of heartbeats by breathing can also be illustrated with sound. On the screen of the computer, a red flash will appear and a beep sound will be produced when the heart beats. As we know, during breathing, interbeat intervals should be changing, so that exhalation will slow the heart down and inhalation will speed it up. Now listen to the beating heart. However, while the last two movies illustrated how coupled systems can influence each other's timescales, this is not synchronization yet. To detect synchronization, we need some quantitative techniques. The first observation of synchronization in a living system seems to be done in a population of yeast cells. It is well known that in yeast cells there can be almost periodic self-sustained oscillations of concentrations of some chemicals. These oscillations can be affected by the level of glucose supplied to the cell. The figure in this slide illustrates what happens when periodically oscillating flux of glucose is added to the population of yeast cells, which themselves oscillate periodically. In all three panels, yeast oscillations are shown by the upper traces and denoted as F, and oscillations of glucose by the lower traces and are denoted as V in. In the first panel from the left, the amplitude of glucose influx is much smaller than the amplitude of yeast oscillations. Moreover, its period is very different, in fact much smaller, than the period of self-sustained oscillations of yeast. As a result, oscillations of yeast are almost unaffected by variations of glucose influx. In the middle panel, the frequency of glucose oscillations is more or less the same as before, but its amplitude is larger. As a consequence, they seem to produce somewhat bigger effect on yeast oscillations by introducing more variability in its instantaneous periods. However, in both these cases, glucose oscillations do not synchronize yeast oscillations, and their instantaneous periods remain very different. In the right-hand panel, glucose amplitude is much larger than before. With this, the period of glucose oscillations is much closer to the period of natural oscillations of yeast, although it still stays smaller. However, even with a relatively small amplitude of glucose, it becomes possible to synchronize yeast cells. One can see this very clearly by comparing instantaneous periods. The periods become equal, and this is called one-to-one synchronization. It is reasonably straightforward to detect synchronization from instantaneous periods. However, not in every case it is possible to introduce such periods. Sometimes the processes involved can have a large degree of randomness in them, and their realizations could look like shown in this figure. It then becomes impossible to sensibly introduce any threshold that the signals could cross, and in this way mark the start of a new oscillatory cycle. Also, there are many processes in which more than one time scale is involved. An example is shown here. This is a signal coming from a system in which there are two time scales. One is faster, one is slower. If we try to extract instantaneous periods, they will represent only one time scale, the faster one, and the second time scale will remain unnoticed. Situations like this require more sophisticated approaches for the detection of time scales. In particular, we need such an approach which would allow one to detect all time scales present in the given oscillatory process reliably. We would like to have a more universal approach that is not too sensitive to the choice of parameters, such as threshold level, which we used for instantaneous periods. Such a method exists, and it is called power spectrum. It works as follows. Suppose we have an oscillatory process, and we assume that it is generally a random process. This is a reasonable assumption because all real systems are affected by random fluctuations, and their processes will always be technically random to a greater or smaller extent. Suppose our recorded signal x of t originates from this random process denoted as capital X of t. Also suppose that we run the same experiment many times and record many signals. Power spectral density, called for brevity power spectrum, can be introduced as follows. Firstly, we take a single realization x of t recorded during an observation time 2 capital T's and find its Fourier transform. This is called finite time Fourier transform. The result is a function which depends on frequency f. Secondly, we find the square of the modulus of the finite time Fourier transform. Because the Fourier transform is a complex function of frequency f, the square of its modulus can be found as the function itself multiplied by its complex conjugate. Thirdly, assuming that we can record all possible realizations of the given random process, we do the same with every realization. After that, we average all squared moduli of the finite time Fourier transforms over the full ensemble of realizations. We obtain the energy of oscillations of the given process at the given frequency during the finite observation time. Fourthly, we introduce the power of the random process at the given frequency f, within the finite observation time. For that we take the energy obtained above and divide it by the observation time itself. This result depends on the observation time 2t of every realization and is not a desirable mathematical result. Fifthly, to obtain a characteristic which describes the total power of the process at the given frequency, we take the power obtained in item 4 and find its limit as the observation time t goes to infinity. Note that when the observation time grows, the energy grows too. So when t is increased, the top and the bottom of the power grow together. However, their ratio could stay a finite quantity. The result of this procedure is the power spectral density, or just power spectrum, of the given oscillatory process. The spectrum depends on the frequency f of oscillations. How should we interpret the power spectrum? In the figure, a sample power spectrum is given for positive frequencies only. Mathematically, the spectrum is an even function of frequency f, so the graph of this function should be symmetric with respect to zero frequency. However, only non-negative frequencies can be easily interpreted, so for practical purposes, the spectrum is usually shown only for non-negative frequencies. Consider a small range of frequencies between f and f plus df. The area under the curve within this range gives us the power inside the given frequency range. Obviously, the power in a spectral peak is larger than the power in the background within the same frequency range, so the spectral peaks indicate the most probable frequencies of the random process. The central frequency of a spectral peak indicates the value of the time scale in the process. There can be several peaks in the spectrum, which indicate several time scales present in the process. The power spectrum is a universal tool to detect all time scales involved in a process if this process is stationary. Here, an example of the power spectrum is shown, which is obtained from a human electrocardiogram. In typical situations, we only have a single realization, a single signal recorded from the given system, such as the heart. In order to obtain the spectrum of the process, we need a collection of many realizations over which we could average power. To achieve this, we take a long realization and split it into many segments and treat every segment as a separate realization. In the spectrum shown, we can clearly see three different time scales. The first one corresponds to the highest spectral peak, whose central frequency is denoted as f(AHR) for average heart rate. This is the peak associated with the repetitive heartbeats. The second frequency is denoted as f(RESP) and points to the time scale associated with respiration or breathing. The third time scale has central frequency denoted as f2 and can have several physiological interpretations which we do not discuss here. Note that around the main peak there are peaks at the so-called combination frequencies, which represent the sum and the difference of the main frequencies. Such peaks are quite common and indicate that there is active nonlinear interaction between the sub-processes with different time scales within the given process. Let us illustrate how synchronization can be detected from experimental data. Here we consider the case when two processes with different time scales interact within a single cardiovascular system of a healthy human who breathes periodically at a certain frequency. In the figure, the upper trace shown by black is an electrocardiogram. Brown curve shows the breathing signal recorded together with the ECG. On the breathing signal, blue circles indicate the moments when the heart beats. If we count the number of heartbeats during a single period of breathing, we discover that there are exactly 3 beats of the heart during any single oscillatory breathing cycle. This is an indication of a 1-to-3 synchronization between the heart and the breathing. Now we show how the same 1-to-3 synchronization can be visible in the power spectrum of only one of interacting signals, whose frequency is the highest. Here the figure shows the spectrum of electrocardiogram, and the human breathing is at the frequency 0.5 Hz. This frequency is very well visible in the spectrum of ECG. At the same time, the frequency of the heartbeats can be seen as the central frequency of the highest spectral peak at 1.5 Hz. The condition for 1-to-3 frequency synchronization between the heart and breathing reads: the frequency of heartbeats should be related to the frequency of breathing as 1-to-3. Indeed, 1.5 divided by 0.5 equals 3. The general condition for N-to-M frequency synchronization between two interacting processes can be formulated as follows. The frequencies of these processes should be related as integer numbers n and m. In other words, the ratio of frequencies should be a rational number. I would like to acknowledge professional and technical assistance from Olga Sosnovtseva and Dmitry Postnov.